Okay, maybe we can start right now. It's a great pleasure to invite you, Jason, to give us this talk. And on behalf of our World Without GNE Myopathy, just to give you an idea about World Without GNE Myopathy, it's an organization, a patient-based organization of GNE myopathy patients. And generally, we represent most of the GNE myopathy patients that have been diagnosed. There are a few hundreds of them that have been diagnosed so far. But we suspect that there may be much more there, given the population and also the social structure that we have. The major goal of this organization is to make clinicians and researchers aware of this particular disease, and also to actually also to work towards finding a cure. I think that's one of the primary goals that we have: find something that will prevent the progression of the disease, or we are hoping that it will at least prevent progression of the disease, if not cure it completely. So that is essentially the main goal of it, and we are looking for all scientific development that is happening in the country. So and your work came to our notice, and I think is one of the really landmark paper that has happened in this field for last few years, and really it points towards a possible way, a possible defect in the GNE myopathy disease, and a method by which we can probably find a cure for it. Dr. Doles has actually done his PhD from MIT, Cambridge, and then postdoc at Barcelona, Spain, as well as University of Colorado at Boulder. And then he's currently at Indiana School of Medicine as an associate professor of anatomy, cell biology, and physiology. And he's involved in multiple other things, including, looking after the PhD program, graduate programs in Mayo Clinic, as well as Indiana School of Medicine. His interest is, current interest is actually looking at various kinds of, genetic disorder affecting muscles. So, Jason, we are really looking forward to your talk, and we really thank you for agreeing to give this talk for WWGM. It's all yours.
Yes, well,
just
again,
1 minute for
ahead.
people who want to ask questions. Put it in chat box, then we can ask him question at the end of the talk. Okay.
Yeah, and I will do my best. I'm not sure how it works with the full screen, but if I see questions pop up, I may just answer them as they or yeah, I mean, either way, I'm not terribly concerned about that. But anyway, yeah, thank you so much for the for the introduction. Our lab has many interests, and certainly our interest in neuromuscular disease is rather new. So we have a lot we can learn from people who've been studying it for a long time, like you all, and we're really looking forward to building more collaborations with people not only in the United States but around the world, especially if there are scientific societies specifically geared towards particular diseases that we've had some experience in. And I apologize, I wasn't able to. I know that the invitation was for to attend the World Without GNE Myopathy meeting, or maybe it was a research day or something, but my calendar was a little busy then. But I hope that the people, obviously, I'm sure the meeting was a little bit bigger than this, but if you see the recording, please do reach out, and I'm happy to Zoom or anything, to chat about some of the work. All right, so I will share my screen. Okay, and hopefully everyone can see that. If not, I'll be talking to myself. But anyway, yes, I am an associate professor, Indiana University School of Medicine here in Indianapolis, in the USA. We've been here only for about 3 months. We've started my lab at the Mayo Clinic in Rochester, Minnesota, and where we initially developed an interest in neuromuscular disease through a very fruitful collaboration we had with a clinical neurologist who we continued to partner with, not only with regarding GNE myopathy, but a number of other rare neuromuscular disorders. Okay, hopefully this works. Okay. So just by way of introduction, I wanted to give people a sense of kind of the work that we did, because it might make sense with the way that we kind of frame questions regarding GNE myopathy. But our lab is really fundamentally interested in muscle regeneration, muscle metabolism, muscle maintenance, really in the context of normal health, aging, and various diseases and trauma-like settings. So many of our studies really do focus on the powerhouse cell type in the muscle stem cell. These are the cells that really drive muscle regeneration, although they don't do it alone, as you can see on the bottom. So can people see my mouse when I do this? Yes, give a thumbs up or whatever, or if not, that's okay. There are a lot of other cell types in the muscle microenvironment that can influence muscle stem cell function, which can thereby impact the ability of muscle to repair and regenerate over time. So while we might, today I'm going to focus mostly on the intrinsic process of myogenesis or muscle stem cell function, there are, and I'll touch on this really at the end, other cell types that we believe or that we know contribute to this process, which we think are not only important in normal muscle regeneration, but could potentially be important in the context of GNE pathophysiology. So we do study muscle regeneration. We do it in the context of several different disease and trauma contexts. We study muscle atrophy, sometimes called cancer cachexia, in cancer patients. We study the ability of muscle to replete and to gain mass back and function back following acute traumas like sepsis. And importantly for today, we are interested in looking at muscle regeneration, although initially it wasn't about muscle regeneration. We were interested in just basic mechanisms of neuromuscular disease, particularly GNE myopathy, but then it ended up being a story that was a little bit more in our area of expertise about stem cells and muscle regeneration, which I'll get into in a minute. So I know that a lot of people here are experts in GNE myopathy, so I was going to keep the background fairly short. But for those who are just joining, who maybe are clinicians or experts in other neuromuscular diseases, I'll just give a little bit of background about GNE myopathy. The good thing is that the causal gene is known. So it is GNE is a gene that encodes a bifunctional protein that has both a functional epimerase domain and a kinase domain, and mutations in GNE occur throughout the gene. So there really isn't a whole lot of rhyme or reason or a critical domain, let's say, that's the most important to develop or that's most susceptible to causing disease. And that's something we're actually interested in studying, is the relationship between different mutations and where they are and how that impacts disease severity. We know that GNE is a key, the protein encoded by GNE is an important mediator of sialic acid metabolism. So in many patients, there are defects in sialic acid production. I think the literature is somewhat divided as to whether or not the lack of sialic acid or the defects in sialic acid metabolism are directly contributing to disease pathology. But I think that's whether or not it's doing it directly via its role in sialic acid metabolism, or perhaps an undefined role in a different metabolic pathway or cellular signaling pathway, remains to be seen, but at least this is what's known so far. Relatively rare. I mean, the numbers somewhat vary, but it's a relatively low number, about 5 in a million, in at least in the United States, and in some patient populations around the world as well. And at least with respect to diagnosis, there's it's really limited right now to clinical muscle histopathology, and in particular, once a suspected case is identified, verified by DNA sequencing. So I'll just share a little bit of the histopathology from 2 GNE patients that were the source material for some of the cells that we used in this particular study. So when I use these acronyms GNE1 with a superscript or 2, that just refers to different patient, completely different biological patients. So not just a clone, we have also multiple clones for each patient when we develop cell lines from them. But as you can see from some of these muscle cross sections, these GNE patients do exhibit do exhibit classic hallmarks of GNE myopathy. They are identified by these positive acid phosphatase stainings. You'll see often, you'll see often staining of these Congo red positive inclusion bodies here in the pictures on the upper left. And they often stain positive for markers of autophagy, including P62. And showing here on the left is just the Sanger sequencing plots showing that once suspected cases are identified via histological analyses, often they're confirmed or validated by DNA sequencing methods. So there are several models of GNE myopathy, that people have used to study and to learn more about what GNE as a gene, as a protein, is actually doing. There are full-on knockout mice, and there are mutations that are designed to represent highly prevalent human mutations as well, including the M712T knock-in mice. Ones where they've also introduced a transgene on a GNE null background to kind of simulate low levels of GNE in combination with a mutated version. The bottom line here is that there are several models that already exist, at least in the context of murine models, and they've taught us a lot. They've taught us a lot about canonical GNE function. They've taught us about the role of GNE mutations during development. They've taught us many aspects about GNE pathophysiology, and and some of them have even served as a resource to be able to study and test some preclinical interventions, including some of the studies where they've tested various forms of sialic acid precursor supplementation as a means to counteract what was thought to be a defects in overall sialic acid levels in some of these patients. Importantly though, while we have learned a lot about the biology of GNE from some of these models, unfortunately the way that they present in mice is very different than the way that we see GNE manifesting in humans. And it's somewhat unfortunate, but it's also an opportunity to learn what we can from these models, but also to move forward and to develop more that better recapitulate aspects of the human disease in ways that maybe these mouse models cannot. So really the big questions that our lab is interested in is, especially since it's not entirely clear that the mechanism of muscle weakness in GNE myopathy is due to defects in sialic acid metabolism, we were kind of interested in taking an unbiased approach to try to better understand the mechanism underlying muscle weakness and dysfunction in GNE myopathy. And then if we had a cellular model, could we target some of either the pathological features? Our idea was that maybe we could target some of the aggregates that were present in some of the GNE patients. We thought that maybe some of the signaling pathways and other things would lead to us down that road. Or just even aberrant signaling to reduce and to counteract, muscle dysfunction related to the GNE mutations. There are a few obstacles though. So there are, I already spoke to the limitations associated with the currently available cell and animal models. And really there aren't a whole lot of, whenever you're studying a new pathway or a new intervention, it's nice to benchmark some of that relative to what is already currently available. But really, there's not a complete consensus around what effective therapies actually are. So it's difficult to sort of benchmark new studies relative to existing ones when there really are not a whole lot of interventions that are clinically effective for the management of GNE myopathy. That said, there's an opportunity for even people, researchers like us who are somewhat on the newcomers to the neuromuscular disease field to use our knowledge and our resources and our expertise that we've gained over the years in other fields, developmental biology, stem cell biology, other things, to partner with some of our clinical colleagues and leverage institutional resources and our existing cell and mouse modeling expertise to have a new take on GNE myopathy. So really our primary objective, which I'll talk about today, is really to advance cellular and what I hope to be better animal models of GNE myopathy that can be leveraged to study disease mechanisms and test novel interventions. So a brief primer, since I will be talking about this cell type for the vast majority of the talk, is this acronym called iPSCs. So it stands for Induced Pluripotent Stem Cells, and these are patient-derived in the case of what we're studying right now. So I'm sure many of you are familiar with it, but in case you aren't, you can take terminally differentiated cells from a patient, and you can use a variety of different cell types. You can use circulating PBMCs, you can use dermal fibroblasts or keratinocytes, hepatocytes, whatever the case may be. So a lot of diverse sources can contribute to can serve as the source material for these induced pluripotent stem cells. And essentially what you can do is you can de-differentiate and turn them almost into an embryonic-like state. So it's very similar to embryonic stem cells, if you're familiar with those. And importantly, what this does is that you now have a cell population that matches the genetic background of the patients that you can then bank and maintain indefinitely. And which is really important actually for rare diseases where you don't often get a lot of patient material coming through the clinic, coming through the lab very often. Now you have a renewable resource of patient-specific material that you can use indefinitely as not only a discovery tool, but also, as I'll just mention in a second, as a tool to potentially screen and look for new therapeutics. So what you can do with these cells, you can certainly maintain them and bank them, but you can differentiate them to a number of different cell types, including in this image, it's showing how you can differentiate iPSCs into neurons or organoid cultures. In our case, we differentiate them a lot down the skeletal muscle lineage. It's actually really neat when you are studying diseases that involve multiple cell types. So if you're thinking about ALS, you could potentially differentiate them down into muscle lineages as well as into motor neuron lineages to be able to study the interactions between different cell types when different mutations are present or absent. And there's a lot of very cool studies that you could potentially do both in the discovery science as well as in the translational space. And there are numerous studies, I couldn't put them all here, that use these now differentiated cell types that you get from this renewable pool here to not only learn more about disease mechanism, but to use these cells potentially for drug screening, for cellular therapies. And if you're really interested in particular patients, you can use them to test novel patient-specific therapies and interventions. So we're not the first people to think about doing this in the context of neuromuscular disease. Here's just an example of a publication in 2017 that used patient-derived induced pluripotent stem cells to model VCP myopathy. So it's not directly related to GNE myopathy by any means, but it just shows that in principle you can generate cells that recapitulate aspects of the human disease, including in this case particular phenotype of aggregation marked here in the upper row with TDP43 staining that they see in the patient lineage, as well as increased accumulation of autophagy markers, including LC3 and P62, as again shown on the right-hand side in the middle and bottom column. So it has been used to learn more about the basic biology of VCP myopathy. I haven't seen a whole lot of papers that have built upon those initial observations, but certainly the potential is there. So really our question was whether or not we could leverage this approach to model and study GNE. Just briefly, I'll take you through an experimental workflow of what we did, and what we did with both in our lab and with the collaborators at the Mayo Clinic. Was that we had a biopsy of dermal fibroblasts, which we received from the patients. We'll start on the workflow on the left here. We reprogrammed these into iPSCs in collaboration with the Center for Regenerative Medicine BioTrust at the Mayo Clinic. We subject them to a number of iPSC quality control metrics. This is important because the process of deriving induced pluripotent stem cells can lead to some genomic instability. So you want to make sure that the karyotype is maintained. You want to make sure that they indeed are fully de-differentiated back into embryonic stem cell into an embryonic stem cell like state, so that they express pluripotency markers. Here's an image of a colony of iPSCs that are stained for various markers of the pluripotent state, so the ability to differentiate, they're still maintaining that undifferentiated state here, so they express markers like OCT4, SSEA, NANOG, etc. And then one of the quality control methods that is frequently used is that we want researchers like to make sure that cell types that are de-differentiated can then at the very least maintain their ability to differentiate to the three main germ layers. So in this case, they maintain the ability to, or we want to check to see if they can differentiate down into the ectoderm, endoderm, and mesoderm lineages and express various markers associated with that. These are typical iPSC quality control steps that are taken, with every clone that we make, with every patient that cells are derived from. And I guess are the main quality control with respect to the phenotype, but also there's a lot of QC with respect to making sure that they're not contaminated and everything before they're banked and maintained for future use. And then really our question was to, our goal was to be able to use GNE patient cells and then to differentiate them down a muscle lineage, and then to be able to study either these cellular myotubes that can form when you differentiate them down the skeletal muscle lineage, or to study them in a context once we are able to understand, or once we differentiate them down a muscle lineage, to be able to then study either physiological or cell signaling differences that may occur between control and patient-derived cells. So another important, quality control step to do that is somewhat taken, sometimes taken for granted a little bit, is to make sure that the patient-derived iPSCs maintain their underlying genomic mutations. This is not always the case. Sometimes there's some epigenetic reprogramming that occurs, and DNA repair and other things that occur that where the mutations are lost. So indeed, we confirmed that with the two patients that we had here, shown here on the left, there's just sequencing confirmation that we were able to maintain the genomic composition of the patients, at least with respect to the mutations associated with the clinical biopsy. And with the GNE gene, we didn't do whole exon sequencing or anything, but they at least maintained the genomic integrity of the GNE locus. And interestingly for us, when we looked at these cell types for, again, canonical markers that are associated with, GNE myopathy, but not necessarily specifically. There are a lot of other, these can also mark a lot of other, or be upregulated in a lot of other contexts of muscle disease as well. But we do see that in two patients, we see accumulation of TDP43 in the GNE patient 1 and patient 2 cells. We see pretty substantial accumulation of LAMP1, which is a marker of autophagy, or a component of autophagy that's typically upregulated either when autophagic flux is increased or autophagic flux is blocked. And I think that's an important distinction I'll get into in a second. And as you'll note here, actually, the phenotype of GNE patient 1, the LAMP1 staining and the severity and the size of the aggregates in this patient were a little bit more severe than in patient 2. And that was very interesting to us, because when we looked back at the patient biopsies, this actually reflected the severity with respect to not only the histopathology, but also the clinical parameters associated with these patients. So were able to take cells from a patient with more severe disease and less severe disease, and we were able to get cell types to derive cells that had a muscle pathophysiology marker phenotype that had more severe markers versus less severe. So that was at least an initial promising sign to us that we were going to have something that we could use as a tool to learn more about the disease. So again, the first thing, as I mentioned, just a few slides ago, was that we wanted to make sure they could maintain germline differentiation capacity, so they can express, all the cell lines express these pluripotency markers, and they all were able to differentiate down the 3 germ lineages. And this is very important, as I'll get into in a second. So this indicates to us that these cells, that GNE patient cells, despite the fact that they have mutations in GNE, don't have an intrinsic defect in just cellular function. Like we don't see proliferation defects or increased rates of apoptosis or anything. Like there's nothing inherently wrong with them, and they don't have any major, at least qualitative, defects in the ability to just undergo cellular differentiation. So they're able to differentiate down the germ lineages just fine. We found that they were equally able to go down an ectoderm, endoderm, and mesoderm lineage, as well as control cells. And the bottom line here is, I don't have a ton of labels on here, but it just shows that there's really no difference between any of the patient cells and any of the GNE cells with respect to both pluripotency markers, as well as their ability to differentiate down the germline. So our next goal was to differentiate these down a skeletal muscle lineage. And actually, before I do that, I'll just go over myogenesis in a nutshell, just so that you can have a sense of what we did and what we're going to be doing. And then, as I alluded to in my title, that we see defects in this process called myogenesis. So I thought it was important to explain it a little bit more. So essentially, that muscle stem cell that I told you earlier was important for fueling the process of muscle regeneration and repair is normally quiescent in adult resting muscles. So it's very active during development. It helps to form new muscle fibers during the course of embryonic development, and it's important for the hypertrophy that happens during adolescence. So that's this process of quiescent cells that are activating and differentiating, et cetera, et cetera. But as an adult, they're typically quiescent. They're little rare spots in the muscle. They only make up about 2 to 5% of the total nuclei in the mature muscle in the mature adult muscle. But they do maintain their ability to wake up. They maintain their ability to be activated upon stimulus. So that stimulus can be trauma. It can be a disease state. It can be even just the normal process of tissue repair and regeneration. So in response to exercise, maybe these cells can activate. And what they do is that they undergo expansion similar to what any stem cell would do. So there's a transit amplifying step where they proliferate into this myoblast pool. Some of these self-renew back to maintain the resting state of the muscle stem cell pool, but the majority then go on to differentiate and either fuse with each other to form new myofibers, or they'll fuse into damaged myofibers to repair any damage. So we know that these cell types are responsible and required for muscle regeneration. There were several landmark papers that came out in around 2010, 2011 that showed that if you genetically ablate these particular cell types and subject muscle to an experimental injury, that muscle is unable to repair. So what you're looking at are just images, cross sections of a tibialis anterior muscle where the left side, the contralateral muscle, was not injured, and the right side was. If you have stem cells, you're able to repair and regenerate muscle just fine. If you deplete them, the muscle is unable to regenerate at all. So just showing genetically and in a physiological context that these satellite cells are important for the process of repair and regeneration. So a little bit more detail into this process of myogenesis. You can see here in just a slightly different schematic of it. Upon receipt of an activation signal, these satellite cells activate and proliferate, they differentiate, et cetera, and importantly, for the context of us studying myogenesis in a dish with patient-derived cells, is that you can recapitulate this process pretty well using either genetic tools that people have done to force cells down a myogenic lineage, or in this case, a cocktail of growth factors and inhibitors that can coax and coax cells down the muscle lineage. So in this case, the iPSC state is actually even a little bit more primitive than satellite cells, because again, remember, these cells can differentiate to many different cell types, including neurons, liver cells, skin cells, anything else. But one of the first things that you do, and what this, several different companies now sell media cocktails where you can differentiate these cells down, and by the time you can get them to a state that actually is very reminiscent of a satellite cell, so that this will be referred to as stage 1 later on in my talk. And then there's various stages that allow us to pinpoint exactly what is happening in these cell types or in the iPSCs at various stages of myogenic differentiation. So we can mimic this process in vitro, I think, is the bottom line here, using a chemically defined cocktail of growth factors and signaling inhibitors. I will be using a lot of markers here, and so I just wanted to have it up so that people could see when we're looking at various stages of myogenic differentiation. So the process of going from a stem cell to a fully differentiated fiber, there's the ordered expression of various transcription factors. So some of the PAX transcription factors like PAX3 and PAX7 generally mark either the quiescent or activated and proliferating myoblast stage before there's really any terminal differentiation into either mature myocytes or terminal myofibers. You'll start to see expression of these transcription factors, the PAX transcription factors, replaced by some of the myogenic factors like MyoD and myogenin, and then later on we'll see expression of different myosin heavy chains that are I think more indicative of late stage different, and not that I think that are indicative of later stages of myogenic differentiation. So when we did this with the patient-derived GNE myopathy cells, we really didn't see a whole lot of difference in the earlier stages, the ability of these cells to differentiate when we are now staining for some of these markers in a dish here, so they're able to differentiate and express markers like MIF5 and myogenin more or less okay as they, as the control counterparts here. So really not a whole lot to see here, except for the fact that one of the more severe cells had a less expression of PAX3, which we haven't been able to exactly find a compelling explanation for, but we find that is actually quite an interesting phenotype, although they are able to catch up later with respect to progression through myogenesis. But perhaps the biggest phenotype that we saw was at the late stage. So when we are finally getting to stage 3 and we're looking for a terminal differentiation marker, so in this case, we're staining for a myosin heavy chain species using a monoclonal antibody that everyone uses, MF20, that stains the multinucleated myotubes and late stage single cell myocytes. What you can see, hopefully appreciate, is that in the control cells, we're able to differentiate the control iPSCs into nice nascent myofibers. There's robust elongated myotubes that are forming that are MF20 myosin heavy chain positive. But unfortunately, when we did this with the patient GNE cells, we either saw very little in the case of patient 2 or almost none at all in GNE patient 1, which was both disappointing, in some ways, because we wanted to be able to fully differentiate these GNE patient cells into muscle so that we could then study it. Because if we can't differentiate them into muscle, we can't really study mechanisms of muscle disease very well, at least in the mature myofiber state. But at the same time, this opened up neat opportunities in the sense that we were able to now see that, okay, well, despite the fact that GNE patient cells can differentiate then, down other germ lineages just fine, they appear to have a specific defect and an inability to terminally differentiate when it comes to skeletal muscle specifications. So with every disappointment comes a new opportunity to be able to think, okay, well, what could this possibly mean? Like, how might this relate to to GNE pathophysiology? Like, we scour the literature and we're trying to understand, okay, well, have defects in myogenesis or muscle regeneration been previously linked to GNE myopathy? And a few papers have mentioned it, but really haven't studied it very much at all. Oh, did someone unmute? Does someone want to ask a question? Or okay, well, whatever. We can get to that at the end if you don't want to type it in the chat. But it was definitely new to us, and at least from the context, at least from the perspective of a stem cell biologist, this was an opportunity to learn more about whether or not there really was a defect in myogenesis and whether or not this could potentially be a contributing factor to GNE pathophysiology. So how might this actually relate based on what's already known about GNE? So I think, the important thing to remember when you're talking about diseases of muscle wasting is that this is really a combined effort between competing forces of muscle growth and muscle breakdown. So if you're thinking about a multinucleated muscle fiber, there are pathways that are constantly and stimuli that are constantly wanting to break down the fiber, because of injury, exercise, whatever. And then those are balanced, in a steady state level by either processes that cause the muscle fiber to intrinsically grow. So these are intrinsic to the myofiber now. So these would be processes of myofiber hypertrophy that I'm specifically talking about here, and that these can together work to maintain muscle mass and function in healthy individuals. Now, importantly, there's another growth or anabolic side to the maintenance of muscle fiber mass, and that involves either when the hypertrophy pathways can't necessarily compete or can't necessarily compensate for the increased breakdown and catabolism, there are, again, these muscle stem cells or progenitor cells that can be activated that can form new source material essentially for these myofibers, either for helping to repair and fuse in to repair damaged pathways, or damaged fibers, or to form new fibers in the case of fibers, whole fibers that are completely lost, either via to injury or disease. So there's and this is I think the really important thing that's often forgotten is that there's two forces that are really balancing muscle mass maintenance in healthy muscle, and that importantly in the context of almost every muscle disease. You really see frank wasting, you see apparent loss of gross loss of muscle function when these two processes become imbalanced. So either muscle catabolism or breakdown becomes a serious problem and outpaces the ability of the cell to repair and regenerate, or you see suppression of growth pathways that lead to a net balance or a net result of there being loss of muscle mass. And I think it's important to consider because that in the earlier stages of a lot of these muscular dystrophies, including Duchenne's muscular dystrophy, you don't actually you see catabolism from a very earlier stage. You see muscle breakdown, defects in dystrophin that are leading to myofiber fragility and defects of the DCG complex early on in life. But there are no, the patients are fine. And that's because of the compensatory action of repair and regeneration pathways, which are able to balance that at least early in life. But by the time adolescence comes around, by the time, this process has been going on for years, eventually the process of repair and regeneration breaks down either via stem cell exhaustion or other kinds of things that eventually cause the balance to tip towards catabolism, and then patients start to have symptoms. You start to see people, you start to see you start to have more frequent falls, et cetera, and it just progresses and gets worse. So our hope is that by studying the biology of muscle stem cells, while we, again, are not really geared towards a direct cure of finding, let's say, a gene therapy to replace GNE myopathy, or exactly replacing the function of loss of function of GNE by supplementing with sialic acid precursors. We believe that muscle progenitors or stem cells are a great therapeutic target because they're a long-term self-renewing population of cells that endogenously exist in the patients. And at least in some preclinical studies in other muscle diseases and scenarios of muscle loss, either with age, sarcopenia, or cachexia, that boosting the regenerative capacity of these endogenous stem cells has shown the ability to delay disease onset or disease or wasting progression in several preclinical models. So we do think that it has the ability to not necessarily cure a disease, but to improve the healthy the health span of individuals who have who do exhibit muscle wasting, whether it's disease or trauma based. All right, so the next thing that we wanted to look at when we were looking and trying to really see if whether or not there were defects in myogenic differentiation was to now take a look, instead of looking at cellular markers, we wanted to look at molecular markers. And one very comprehensive tool and unbiased tool, relatively unbiased tool to look at global changes that are happening on a molecular level, at least with respect to RNA and transcripts, is single cell RNA sequencing. So just a brief primer to that for those who are maybe not familiar with single cell RNA sequencing work is that this essentially allows us to look at a heterogeneous tissue or a heterogeneous population of cells to be able to process all the transcripts in each cell individually, because these single cells are now barcoded. We can take the transcripts, we can sequence them, and then we can reassign them back to individual cells. So, and then the great thing here is that you can not only see differences between different cell types, but then you can look at gene expression within those cell types and compare them to others in that given experiment, or even similar experiments that were run with the same technology. So it gives you two things. It basically gives you the ability to do transcript profiling, which people have been able to do for years, either with conventional RNA sequencing or with microarrays back in the day. But it allows you to now do this at single cell resolution to be able to look at all the cells in a given tissue. And now people do it with nuclei, they do it with a whole bunch of other things, but I'm not going to get into any of that right here. So basically what we did was we wanted to, because we didn't have any assumptions necessarily that even though these cells are a cell line and that they're iPSCs, we weren't going to make any presumptions that they were all behaving in the same way as they progress through in vitro myogenic differentiation. So what we did was that we compared control and patient cells from different stages to test that hypothesis, to see how either maybe some subpopulations or other cell types were moving through myogenesis, and to see at what point did they start to separate from the controls. At what point do GNE patient cells start to separate from the control cells? Because we got a sense of that from some of the cellular experiments, but we didn't really get a great sense of the precise timing of when differentiation is impaired, nor do we really understand the molecular underpinnings of that defect. So what I'm showing you here is a two-dimensional representation of some of those single cell sequencing data. So what this basically does is this plot is showing you it condenses down all of the multidimensional complex information of the entire transcriptome of a single cell, and it places it in two-dimensional space. So what you're basically, cells that are closer to each other are more similar on a global transcriptional profiling level, and those that are farther away from each other are more different, is I think all you really need to know here. So what you can see here is that we're looking at all the cells, both control and GNE, that as they're undergoing myogenic differentiation. So if you focus for a second on the control cells, so the blue ones, as you go from light blue to less, slightly less blue to dark, or to slightly more blue to dark blue, you can see that, cells at an earlier stage are very distinct as they go through stage 1 or stage 2, and that by the time they're more mature, so at this later stage here, this late myogenic precursor cell, they're very different from each other. So clear separations, indicative of the fact that they're undergoing a major cellular transition. However, when you look at the GNE cells, you can see that at earlier time points, they look reasonably, there are differences between the control populations and the GNE populations at both the earlier stage, at the iPSC stage, as well as the early differentiation stage here. They're separated, but they're somewhat similar in this full two-dimensional plot. But what they are unable to do now is to progress beyond this first stage here, when they're in that satellite like cell state or early muscle stem cell precursors. So they're unable to fully differentiate. Now they're trying. You can see the dark red is trying, it's a little bit separated from the time point one, but really it's unable to transition fully to the later stages of myogenic differentiation. So we can see that when we take a look at some of those markers that I showed you earlier, like some of the PAX transcription factors, some of the myogenic specification factors like MyoD and MIF5, MIF6, and some of the later stages like Desmond. So what we can do is I'm showing you two-dimensional plots where we're looking at all the different cell types here, and what we're showing, all the cells on a single plot, and what you're seeing is that expression of the early myogenic factors like PAX3 come on relatively early, relative compared to the other markers. So this is the iPSC stage where there's no expression of a muscle marker. That's good. We start to see it come up, and then we start to see expression of PAX3 go down as the cells terminally differentiate into myocytes and then fuse into myofibers. You start to see MIF5 coming up at that intermediate stage and really kind of at the later stage here. And then even later, you start to see expression of some of the later myogenic differentiation factors. We can display that sort of in, I'm showing you here in dot plot fashion. You can see in the control cells, they express, now we're kind of arraying just a few representative markers from less differentiated to more differentiated here. So as you can see, the control cells start off less differentiated at time point one, they're expressing PAX3, very indicative of that, satellite cell or early proliferative stage. By the time you get to time point two, they're now expressing some of the later markers. Both control cell lines do this. It's not just one. We have multiple control lines now where we do this. It's not just a feature of cells that have muscle disease that they all have myogenic defects. So if you look at GNE, we can see that again they sort of express earlier stages of these myogenic markers, but then do not express the later stages like the controls. But a similar, another muscle disease that we're studying in this case, NMLE myopathy, driven by mutations in ACTA1, doesn't have a myogenic defect in this case. It fully expresses PAX3 at the intermediate stage and expresses all the late stage markers just fine. So this is not just a universal feature. Muscle myogenesis issues aren't just a universal feature of a myopathy, but rather appears to be somewhat specific to GNE myopathy. It might not be limited to GNE myopathy, but in this case, it's not just an artifact of the fact that we're studying something that is related to muscle disease. Okay, so since time point two was the most severe, we wanted to see potentially what were the signaling components underlying that separation or the inability of those cells to transition from time point one to time point two. So we took some of the differentially expressed genes, the differentially expressed transcripts at time point two. We did pathway analyses, and more or less the bottom line is what we saw is that the top 14 of the 16 top pathways were related either directly or indirectly to the integrated stress response. So clearly these cells are stressed. They're stressed in a number of different ways. But what there appears to be is a coordinated, sorry. Okay, could just be background of someone that unmuted. But either way, sorry, there's a clear integrated stress response. And actually, I won't get into it here, but we are very much interested in whether or not this is something that's also targetable, from a GNE myopathy standpoint, and potentially could be targetable in a number of other myopathies where the ISR is a clear, has a clear molecular signature. So we know that, as a component of the integrated stress response, we noticed defects in autophagy. So if you recall back when I showed you the histopathological features of GNE myopathy earlier, we saw accumulation of TDP43. We saw, pretty, substantial accumulation of LAMP1. Here you can see in the patient biopsies themselves, increased expression of P32 relative. Actually, I'm not showing a patient sample here, but normal muscle doesn't stain, is not very reactive to P62 at all. Oh boy, maybe I should use my mouse. Maybe that'll be a little bit less on my keyboard. So one of the top pathways that we saw when we looked at the GNE time point 2 and kind of looked back and forth were defects in autophagy. And we know that defects in autophagy can contribute to the integrated stress response overall. And we thought that it was interesting from several standpoints, but I'll just fast forward a little bit since we're, I don't want to take all day here, is that we did notice when we took a closer look at autophagy in the control versus the GNE patient cells, that we saw, again, things that were very similar to what people had seen before, was accumulation of these markers that were associated with autophagy, like an increased ratio of LC3B2 to 1, increased expression of LAMP1, P62. And a lot of people in the past had assumed that this meant that there was too much autophagy, that there was excessive autophagy, because that is one potential interpretation of it. But when we consulted with some autophagy experts, at our institution, they also noted that this, even though you see increased abundance of some of these factors, that this could mean there's excessive autophagy, but it could also mean that there's impaired autophagy. In this case, autophagic flux that's impaired, that's leading to aberrant accumulation of some of these factors. So we tested that hypothesis. We used a compound that's typically used to block autophagic flux. And what happens in control cells, you'll see here in this graph, when you look at LAMP1, that we, typically when you block autophagy, you'll see accumulation here of, a factor like LAMP1. If you look at some of the patient cells, when you treat with the same drug, you don't see the accumulation now of LAMP1, suggesting that, that autophagic flux is already blocked, and that it's not able to be blocked further with a drug that's known to inhibit autophagy. So our data, at least preliminarily, implicate potentially defects in autolysosome formation and maturation, and a potential block in autophagic flux in patient cells. As opposed to there being hyperactive autophagy that needs to be limited, really. So when we, in parallel to, trying to define the mechanism and trying to understand more about autophagy defects in these cells, we performed another analysis where we're able to take the global transcriptional profile of these cells and then predict what might be able to reverse the, these transcriptional defects. And one of the top factors that one of the top drug candidates that we got from this in silico screen or prediction program was SB203-580, which, if you recall, I was talking about the integrated stress response, is a known inhibitor of PI3 MAP kinase, which, if there are some P38 MAP kinase people out there, is a stress activated kinase that's important, that's activated in a lot of pathological contexts as well. So we thought, okay, it has a role in the ISR. It could be interesting, and we could test to see if it actually is able to relieve some of the myogenic block that we see in these GNE cells. The bottom line is that it did. So when we treated with increasing doses of this drug, SB, we saw the ability of these cells, and you can just focus here on the graph on the right, at even at lower dose, it kind of plateaued after a while, but we saw increased myogenesis in the presence of this putative P38 inhibitor. But then when we checked levels of phospho-P38 and actually looked at P38 signaling, we saw no difference in our patient and control cells, which kind of puzzled us for a second, until we dug down into the literature a little bit more and found out that this drug can also serve as an activator of autophagy. So it does not appear then that SB is potentiating myogenesis or relieving the myogenic block in the GNE cells due to its function as a P38 inhibitor, but rather potentially as its role as an autophagy activator. We're currently in the process right now of testing other autophagy activators to see if they can rescue the GNE-associated myogenic defects. So lastly, I just kind of wanted to, a visual representation of what we're hoping that we can do, is that we hope that by restoring or boosting autophagic flux in the skeletal muscle and progenitor cells, that we can now enhance the growth and repair capacity of GNE myopathy to be able to better balance the catabolic effects, which, again, we're not sure. Maybe the catabolic effects are, that are present, or the the phenotype in mature muscle fibers is related to autophagy or the integrated stress response, but we don't know that right now. We're interested in studying that more for sure. But at the very least, in the meantime, we hope that we can impair the endogenous suppression of these muscle progenitor cells by improving autophagic flux and thereby kind of tipping the balance back in favor of muscle repair over muscle breakdown. So in summary, there really are no effective therapies for many neuromuscular diseases, including GNE myopathy. We do need better models to identify, study, and advance clinically relevant interventions. We believe that we've developed, cells are cells. They're not patient samples. They're not mature muscle. But we believe that we have a cellular model that is importantly derived from human patients. So it's a human cell line. It's not mouse or animal model based that exhibit myogenic defects. We are in the process of characterizing these integrated stress response and aberrant autophagy phenotypes that are present in these cells. And we hope that enhancing autophagic flux may be a viable therapeutic option to boost repair and regeneration in GNE myopathy. So I'll really just close with a few of what I think are some interesting next steps that we want to take our research into. Certainly there are more, but just wanted to highlight a couple. One interesting question that we've had was, why, since GNE is mutated in multiple cell types besides just muscle cell types in patients, is why GNE mutation or loss of function specifically results in a muscle phenotype. Again, we don't know the answer to that, but it's possible that GNE mutation or loss of function in non-muscle cells in the muscle microenvironment could be contributing to impaired tissue regeneration on top of the observed myogenic defects that we see that are intrinsically present in muscle stem cells. So remember, I showed you this image earlier. Even though satellite cells and muscle the muscle cells are the ones that are expanding and differentiating and fusing into muscle fibers, there are a number of other cells besides the satellite cells that can influence muscle stem cell behavior, including fibroblasts, including immune cells, including endothelial cells or pericytes. And when we take a look at some of our just other profiling studies where we've looked at cell types in other mononuclear cell types in the muscle microenvironment, including macrophages, neutrophils, other immune cell types and fibroblasts and such, we do see robust GNE expression in healthy, this is non-patient and non-pathologic muscle, but we do see that GNE is present in a number of in several other cell types, including endothelial cells and fibroblasts, leading us to believe potentially that GNE mutations or impaired function could be contributing to overall muscle regenerative defects in muscle. When we stain for the presence of sialic acid using various lectins in cross sections of muscle, we also see that as opposed to localizing to the muscle fibers or satellite cells themselves, we actually see the most robust staining in the non muscle components of skeletal muscle. So in nerve bundles, we see it expressed also in this image. We haven't done the co-localization studies yet, but these are very similar to what you would see in microcapillaries in muscle around the endothelial lining and the vasculature. So we're really interested in exploring what the potential contributions are, maybe if not, grossly a vascular phenotype, but potentially subtle defects in some of these other cell types might be having that together, can lead to impaired muscle function and gross loss of muscle mass and strength. And second, it really is our goal to take these beyond cells, because again, cells in a dish, even if you can take them down a myogenic lineage, can really only go so far. They can only go into nascent myotubes, which again are not really fully contractile muscle fibers. So we are developing and testing a system that has been explored in the context of embryonic stem cells, and people are now using it for induced pluripotent stem cells, where we can essentially transplant patient-derived iPSC cells into immunodeficient, hind limb irradiated to now deplete the satellite cells in endogenous response, but we can get these cells to incorporate into mouse muscle to form a human mouse chimera, if you will, where the cells that are transplanted now carry the mutations that are present in the transplanted lines. So What I mean by that is that we can have potentially a system where we take cells from a patient, we can inject, we can study them certainly in a dish, but potentially in the mouse as well, and then be able to use that knowledge to be able to either test patient-specific therapies or to learn more about the disease in general. A lot of people talk about bench to bedside research, but what we see here is as a bedside to bench to bedside discovery, translation, research pipeline where we could have, patient specific cells banked and available both as a resource, but also as a way to test potential therapies for rare diseases that might not otherwise have samples coming along very often. When we've done some preliminary transplantation studies with these, you can see here is that when we have a cross section now of murine muscle where we've transplanted some of these cells, they're staining positive. In the left-hand column here for laminin, you can see that stains, the individual muscle fibers in a cross section. When you stain for use an antibody that's specific for the human form of dystrophin, if you're not transplanting any cells, you don't see anything. But when you transplant or when you stain for human cells that have been transplanted to mouse muscle, we see robust staining of human dystrophin, which again, in this blow up here, we can see is localizing to the proper place along the periphery of the muscle fibers. So we're hopeful that we have, a system in which we can start to explore, trying to actually recapitulate, disease, patient GNE cells in a more physiologically relevant context to supplement some of our cell line based experiments. So with that, I'll end. I know it's a little bit past the hour, but I thank you for your attention. A lot of this work was driven by a very talented fellow, now research scientist here at IU, Becky Schmidt. She's actually on this call. So if you have any questions, you can direct them all to her. Sorry, Becky. No, it's fine. But yeah, she was the main driver behind this project, and along with a talented postbac student who's now a medical student at Mayo Clinic. Really want to highlight our collaborators, the clinical neurologist Margarita Maloney, several individuals in the Center for Regenerative Medicine at Mayo Clinic, and a friend and colleague, Gina Rezidlo, who is an expert in autophagy. Number of funding sources that have supported this work, and thank you for your attention. I'm happy to take any questions.
Thank you. Thank you very much. I mean, it's an amazing talk. We can see that we're slowly beginning to understand the biology of this disease. So I guess we are a little bit still have to probably cross a few more hurdles before we are very sure about it. Let's see. Any questions anybody has? I don't see anything in the chat. Okay. The question is from Sudha. Sudha, you want to ask question?
Yeah.
Yeah, no, oh, sorry, go ahead.
Hi, Jason. Thanks for the very wonderful talk. I have a few questions. So, GNE myopathy is an adult onset disease.
Mm-hmm.
And we don't understand exactly why that is so. So do you think that one of the reasons could be the exhaustion of the stem cell population with age, and then that's why it is adult onset?
Yeah, no, that's a great question, and I of course, just since I really do enjoy stem cell biology and I see these kinds of things, these kinds of phenotypes popping up in other kinds of contexts, it does feel like a stem cell exhaustion kind of situation. Now, I don't want to say that it's a number one driver of it, but I think it could certainly be a potential contributor to the adult onset of GNM. But the one thing that I will say is that when people have looked at, let's say, just taken patient biopsies and stained for satellite cells and everything, the numbers are actually still more or less there. Like, you don't see like massive depletion of PAX7 or PAX3 positive satellite cells in GNE muscle, but as you point out, it doesn't necessarily mean that even though they're there, that they're functional. So people haven't done a whole lot of, I think, functional studies, either isolating them from symptomatic patients, from biopsies and studying them further, or, in corresponding mouse models, isolating the satellite cells and actually seeing if they're functional. Those kinds of studies would be certainly something that we actually have planned to do in a related study, but I think could be a potential. I think you're on the right page, in terms of whether or not stem cell exhaustion is a phenotype, because just because the cells are, people have stained and said, well, it can't be, there's obviously no defects in myogenesis or anything else, because there's still, when we count stem cells, they're still there. But yeah, I think we're more interested. I think we're definitely interested in examining them. If we do have archived biopsies, which we do, of some of the patient samples, what we can do is that we can do, from frozen samples, isolate nuclei, and a lot of people can now, and to be able to look then at the molecular profile to get a sense of whether or not stem cell exhaustion, pathways or processes are up in either the stem cells, or either if other cell types are aberrantly activated, like fibroblasts that are suppressing the function of satellite cells. So great question. We're only at the beginning of understanding that, but I would certainly think that there's a little bit of a contribution there to disease onset from stem cell functional deficits, basically.
Thanks, Jason. I have one more question.
Sure.
Your data indicates that the one of the problems could be the autophagy, impairment in GNE myopathy.
Yes.
So just want to know, since I don't know much about this field,
Is the feed frozen for other people?
What is the scenario about
maybe switch off your video because we can't hear you.
Okay, it's not just me.
Oh, okay. I'm sorry. Could you?
If you switch off your video, or at the worst case scenario, you could type it in the chat.
Okay. Can you hear me now?
Oh, that's better.
Okay. Just wanted to ask you whether autophagy related molecules are there already being tried for other drugs, and whether we could hope to try them for GNE myopathy.
Yeah, no, one of the things that we want to do is to look at autophagy activators that are already, granted, I haven't looked for ones that have been approved in other countries, but we are looking for ones that have been at least in, preliminary FDA approval for, I think the most common one are ones that have been approved for cancer indications. But we are at autophagy activators, either ones that are preclinical or ones that have been at least received preliminary approval, but we haven't really gone down that route yet. But the goal would be to start with those that might have the fastest timeline to be able to get, if effective, to have, rapid approval, because they're already, at least preliminary tox studies have been done, and, safety and efficacy. So that would be the goal.
Okay, so
thank you.
The next question is from Arka Ghosh, and he said, could you comment on the differentiation deficit in vitro observed in the patient lines, whereas the patient did develop muscles and the disease is adult onset?
Yeah. So some of our differentiation data would indicate that maybe if these mutations are pretty severe, that they might not be able to develop muscles at all in the first place. So that's a very good question. There's the one thing that we don't know is that, again, we're looking at cell types that are in an isolated context. So if there are other factors that are promoting muscle development that are able to get those cells over the hump such that people are able to develop muscles just fine. In vivo, that could be a possibility. We don't really know if there are other compensatory muscle stem cell types, especially during development, that are a lot of people talk about these fibroadipose progenitor cells that have some satellite cell muscle regenerative capacity in adult muscle, but they're certainly more myogenic in embryonic stages. So I'm guessing there's either cellular, like functional compensation from related cell types during development that allow muscle to develop just fine. And that once the muscle is formed and that the muscle becomes more dependent on specifically adult muscle stem cells, then that's when the issues become a little bit more pronounced. Developmental processes tend to have a lot more overlapping or compensatory mechanisms to allow the formation of a tissue to occur. So I mean, I'm guessing that's the case. We'll be able to get a sense of that a little bit more when we start transplanting some of the GNE patient cells into muscle, because in some ways that recapitulates, kind of the big, the robust formation of muscle and fusion events that occur during development. So if those cells fuse poorly, we'd have, another question on our hands as to why that's the case. But we'll see. I think that's a great question. I saw a question about sialylation assays performed on control and GNE lines. We did. I didn't show those data, but we actually didn't see any differences in sialylation sorry, I don't think we looked at sialylation, but we did look at overall sialic acid levels, and we didn't see any difference between those patient lines, which again was a little bit surprising to us. But we think, at least in terms of the severity of the myogenic defect, the only correlation that we saw was with defects in autophagy and not defects in overall levels of sialic acid. But we should take a closer look, because we haven't, one of the things we wanted to do was to look at the protein sialylome to be able to better understand specific proteins that might be hyposialylated in the context of GNE mutations. And one question, oh, you're welcome. Another question that was,
Ram,
yeah,
Ram wants to ask a question. That's what he said. He didn't write the question.
Yeah, I was unable to raise hand here. So yeah. Hi, Jason. It was a very nice talk. So you mentioned about activating endogenous satellite cells as a therapeutic avenue. From your study, is there any sort of direction that you would like to go in activating satellite cells in this context?
Well, I think the first major hurdle that we have is to be able to establish an in vivo model of this. So to be able to get these cells into mice, to be able to study this in a more physiological context. And again, I'd like to know the answer to the question that was raised earlier, in that whether or not satellite cells exhibit functional deficits in symptomatic muscle. So I think that would probably be step one. Step two then would be to then actually look at to see if we can get these cells into mice. But certainly, yeah, no, I mean, I there's always so many things to do, and so and especially because this is a very small field, there's not a whole lot of either resources available, when we try to get funding, foundation support, or the government support for these kinds of studies. Some people say, oh, it's too rare, we don't want to give you money for it. But the goal is to try to work at least with some internal sources. And I mean, the NIH, so the National Funding Agency for the United States, is actually, I think, had a renewed interest in rare disease biology. So I'm hopeful that we can, get some of the resources to be able to take some of these studies further, because yeah, I do really think that once we implicate, stem cell dysfunction as actually existing in symptomatic muscle, then we have the full rationale to be able to move forward. But I really wouldn't want to push forward until we've 100% established that, because I wouldn't want to waste resources studying something that could just be an artifact of in vitro culture. You really want to be sure before you do that. So I feel like we're on the path, but there's still a number of other key things to do first.
Yeah, I do recall at least one report where primary myoblasts derived from GNE myopathy patients are able to differentiate the IPS-derived ones, which show this defect that you demonstrate. So
yeah, we don't. Yeah, I mean, I do remember that study. I thought those were derived. I thought those satellite cells were derived from a mouse model. Was it patient derived?
I think there's patient derived.
I've seen. Okay. You know, there's a lot of things that can happen when you take cells out of their satellite cells out of their native environment and try to do things like, if there were environmental related suppression of their function, that would not be an issue in cell cultures. So I do think, I mean, our data would indicate that there are intrinsic defects in satellite cells, but it's possible that in some patients it's not as prominent, so that they're able to differentiate just fine, or that at least when they transition from a very, you know, kind of low oxygen, you know, happy in vivo situation, and you put them in a dish with tons of FBS and serum and stuff like that, that they're able to proliferate just fine. At least some of the studies that I've seen really have only looked at like expansion and basic differentiation. So I wouldn't be surprised if you gave, you know, if those cells did have some defects, or, you know, depending on the severity of the patient, which with those cells were derived, that there might be other phenotypes. But, you know, I think that's kind of the point, is that there's really only a handful of studies that have been done so far, and that we really need, you know, more and more patients and cells, you know, to be collected and to be studied in order to be sure about anything, you know, to be sure about sialic acid supplementation, to be sure about regenerative defects. And that's one of the biggest challenges, is just the lack of tissues and cells and everything that, you know, researchers have, you know, at their disposal. But there was one question. Sorry, I hope that kind of answers your.
Sure. If I may, I have one more quick question.
Sure.
So the integrated stress response deregulation that you see is in the time point 2.
Mostly, we do see it slightly at earlier time points, but it's not as robust. And I don't, is Becky on this call? I'm not sure we see any statistical evidence of it at the iPSC stage, like that, the undifferentiated stage. Like, they're not just stressed to begin with.
Is there something that jumps out in time point 1? Because defect in time point 1 is probably the one that's preventing it from going to time point 2.
It could be, yeah. We haven't taken a closer look in seeing, especially with the you know, the precise timing of these the ISR transcription factors. That's something we actually think is going to be better resolved by actually doing, you know, just more classic PCR Western blot analysis of EIF2, ATF4, you know, all the other stress-related factors, and looking to see when they come. Because you're right, at the time in which we're seeing it at time point 2, we're seeing it, you know, at its worst. We're seeing all the stress-related pathways activated, but you know, is it defects in GCN2 or PERK or something that are, you know, in the EIF2 pathway that are initiating the events at time point 1 that are causing the defects? We don't know that, but yeah, we're actually working with an ISR lab here. We're hoping to partner with them soon to get some of their inhibitors and tools to be able to not only look at the time course of ISR activation during differentiation, but then really to kind of probe different, you know, aspects of that at different time points as well. So we don't have that. Yeah, we don't know that right now, but that's something we want to know. Yeah.
Thank you.
Rajesh, you want to ask it, or just his question?
Yeah. Yeah. Hi, Jason. Great talk. I wanted to ask one question. Can you hear me?
Yeah, go ahead.
Yeah, I wanted to ask whether sialic acid analogs, whether they allow the TP1 to TP10.2 differentiation of the defect which you are seeing. Would the sialic acid analogs help it?
We haven't tested those, you know, and I think that, you know, earlier when I was talking about benchmarking other kinds of novel therapies to ones that have had some kind of at least preclinical efficacy, we do want to do that. We haven't done it yet. I think, you know, I think that would be, we were initially going to do that right away, but then when we got the sort of negative data of there being differences in sialic acid levels between the control and the patient cells, then we sort of put that on a back burner. But just because we don't see global differences in sialic acid levels with the one assay that we did doesn't mean that sialic acid analogs might not be able to rescue the the transition from time point 1 to time point 2. But it just became less of a priority once we didn't see differences in global sialic acid levels. But we certainly should test it for sure, officially. And your second question of why distal muscles get affected first in GNE myopathy, I have no idea. I think that the specific muscle group presentation of various myopathy, various muscle diseases is a problem that everyone's interested in. I mean, like as a muscle biologist, I'm very much interested in differences between anatomical differences in skeletal muscle location and how they are susceptible to different diseases versus not. It's very obvious, or there's an easier explanation when they're derived from different germ layers. It's like, okay, well, all the extraocular muscles might behave differently, because they're derived from a different, source. But at least with respect to distal versus proximal and like the leg, for instance, it's not, I don't, I really think, some people have said, okay, neuromuscular innervation or other kinds of things, or people have looked at even satellite cell densities and other kinds of, but I really think at the very best, people have maybe found some correlations, but why a GNE, why defects in, mutations in GNE or defects in sialic acid metabolism might affect distal versus proximal more. Yeah, I don't know that, but it's definitely something that we have in the back of our minds for sure.
Jason, just a continuation on the point. If you find that, because in clinical trials, the sialic acid analogs are not working as they were expected to, right? So is there any possibility of GNE having some altered function altogether with respect to like the main function which is implicated for GNE myopathy is something altogether different?
Well, that's kind of the idea, and which is kind of why we did the unbiased profiling, was because we were, we weren't locked into this idea that it was because of defects in sialation or defects in sialic acid metabolism. We do think it could be moonlighting potentially as another, with another function in either a related metabolic pathway or another signaling pathway or something else. And, I think first we want to, to start collecting and developing tools to be able to, overexpress and better model, do some protein biochemistry to better understand that. But, in the meantime, some of our expression data would indicate that it's not really a metabolic related factor, but somehow related with stress response. But I do think that we need to keep in mind that the phenotype might not be related to defects in sialic acid metabolism per se.
And Jason, one last question. Have you tried rapamycin for like?
In progress. It's actually, we're actually, yeah, that's one of the ones we're studying.
Yeah, great. Thanks. Would love to see your publication on that.
Yeah, thank you. And I'd love to, for people who are interested, we're more than happy, very much interested in collaborating or sharing data. We have no problem doing that. For some physical resources, we did generate these cells at my former institution. So, for resources like that, we have to get in touch with Mayo Clinic and our clinical collaborator, but it hasn't been the problem. We've already shared it with, a couple other folks so far, or at least in the process of doing it. So
Jason, we'll talk to you about future collaboration later.
Okay.
That's a great idea that great for you to offer the resources that you have. I think we'll talk about collaboration sometime. I have some small questions that you have. Is it control our gene corrected one? Patient cells, and what are the controls from your paper?
So the controls are just age and sex match. So this was before these studies were conducted before it became, I think, more required that we use isogenic controls. So we are moving forward. All of our studies are going to be done with either correcting patient mutations or introducing GNE specific mutations de novo into naive cells. So these in particular ones were, there were separate patient controls. So they, like patients derived from healthy individuals. So not a pure isogenic control, which is why we do want to do those isogenic studies before.
Because,
Yep.
If you correct one of, both your mutations are actually compound heterozygous. If you correct one of them, I think that should be because heterozygous situation, these mutations or these alleles don't actually cause any problem. That's what we have seen. And one of your
Also, if you
Indian
correct
origin?
one, sorry.
Is one of your patient of Indian origin?
I'll have to double check that.
No.
But yeah.
Because from the mutation, we feel that it may be.
Yeah, it could be. Yeah.
Okay, my second question is related to the fact that I'm not aware of it, but is nobody, as far as I know, nobody has done a single cell transcriptomics from patient tissues.
Nope. As far as I've.
And are you planning anything like that? Because when you're looking at bulk tissues, you probably may not be able to see the heterogeneity in cell types, and you may find some cells which are showing similar kind of pattern as your single cells, stem cells.
Oh, sorry. I think I might have dropped off for a second. Are you there?
No, I'm sorry. The internet connectivity is always an issue.
Okay. Well, either way, we can connect later. No problem. But again, I appreciate the invitation.
Yeah. There's just one comment. A few days back, I saw a paper where people have done single cell transcriptomics, keeping a cell alive and functionally active. So you can actually follow the same cell through the whole process of differentiation without actually killing it.
Yeah, if you wouldn't mind sharing that with me.
I've shared that with you.
Sure. Yeah. Yeah, I think that would be very powerful to really be able to understand whether or not, these cells retain functionality in pathologic muscle.
Yeah. Okay, thank you, Jason, very much. And I think we'll be in touch with you and we can talk about probable collaborative.
Sounds good. Thank you.
Looks
Thanks.
great.
Thanks a lot.
Thank you.
Take
Thanks
care.
a lot, Jason.
Bye.
Bye.
Bye.